Hi everyone, I'm Sumana Sundaramurthy. I'm a technical account manager at Sino Biological. First of all, I would like to thank all the organizers for giving me the opportunity to present. Today, I'm going to be talking about AI-based antibody discovery service, which could support de novo antibody design as well as antibody affinity maturation. So for people who don't know Sino Biological, we are a one-stop reagent and service provider. We have several antibodies, proteins, as well as ELISA kits on our catalog. Besides our high-quality reagents, we also have several CRO platforms which we use to do customized projects. So getting into the reason why I'm here today, I'll briefly go over the overview of antibody development and then move on to the antibody discovery platforms. And then I'll talk about how AI-based antibody discovery service at Sino Biological works. So looking at the antibody development, when we look at the antibody development timeline, hybridoma technology is one of the classical approaches of creating hybridomas. It came into existence around 1975. And about 10 years after that, we had the phage display approach that came into existence. The first mouse monoclonal antibody, which was approved by the FDA, was around 1986. And soon after that, there came the existence of chimeric antibodies. It's not pretty far away from the chimeric, there came the humanized antibodies. And then in 2000, we had the first ADC that was approved by the FDA. Since then, we've had a number of antibodies, monoclonal antibodies, which have been approved by the FDA for different applications for different diseases. And we should thank the development of technologies, especially in the antibody development platforms, which has come into existence. So when we are looking at the applications of antibodies, you did see in the previous slide how antibodies could be used for therapeutic purposes. Besides for therapeutic purposes, it can also be used for diagnostics. And it can also be used for biomedical research, where we could do multiple assays like Western blot, FACS, or ELISA using antibodies. So when we're looking at the evolution of antibodies, what would be considered as a first-generation antibody are mouse-derived monoclonal antibodies. When we talk about second-generation antibodies, that would be chimeric and humanized monoclonal antibodies. If you're looking at the third generation, it would be the full human mAb, and the fourth would be natural full human mAbs. So next we'll be looking at the different antibody discovery platforms. So the antibody discovery platforms, it is shocking but still true that hybridomas, the classical approach of developing monoclonal antibodies, is still being used. And apart from that, we have phage display platforms, which is also used for developing antibodies. And we also have a single B cell platforms where different approaches are being used to sort the B cells and then isolate the B cells, which would ideally bind to the target, which is being injected into the animal. So at Sino Biological, we do have these three different platforms. So this is actually a schematic of how the antibody development process at Sino Biological works. So let's say if we are looking at the hybridoma platform, once we immunize the mice and the mice achieves the fusion standard, which is actually determined by looking at the ELISA titer values, we sacrifice the mice, take the spleen out, and then isolate the B cells. And those B cells are then fused with cancer cells to create hybridomas, and then they are subjected to screening. So during this process of screening, what we basically do is we isolate the positive clones. So we are isolating the primary positive clones, which would bind to the target. So the primary positive clones would then be subjected to 2 to 3 rounds of limiting dilutions. So during the limiting dilution stage, what we are trying to basically do is isolate the monoclones. So once we isolate the clones, we do another round of validation using ELISA, and then we further expand those clones to produce the purified antibody. When we are looking at the phage display platform, so in this case, we use rabbits for immunization. So once the animal achieves the standard, we collect the tissue, which is the spleen in this case as well, and then isolate the B cells. And from the B cells, what we do is we extract the mRNA, which is then converted to a cDNA. And that cDNA is then used to build up an scFv library. And that scFv library is then subjected to panning, where we are actually trying to isolate the hits, which would bind to the target. And once those hits are obtained, we would then express those scFvs in the recombinant version as a full-length IgG. So here would be a recombinant antibody. So when we are talking about the single B cell at Sino Biological, we can use both mouse and rabbits for this particular platform. So we have 2 different approaches for sorting the B cells. So we can use either FACS or even Beacon for sorting the B cells. So over here, basically, we are sorting the B cells, and then we isolate the B cells that could bind to the particular target. And we are basically extracting the mRNA, and that mRNA converted to cDNA. cDNA is then directly converted into a full-length IgG and expressed in our antibody expression platform. So this is a comparison between a different platform. So this table is one of my favorite tables, where you can actually see the different platforms which we use for developing monoclonal antibodies. We have the phage display here, the hybridomas, the single B cell where we use FACS, and single B cell with the Beacon platform, which is being used to sort. So this table kind of gives you guys like an overview of how, like for example, the duration would take for developing the antibodies using each of these platforms, and also some other important aspects, like for example, VHVL pairing, where there could be mispairing that could occur when we use the phage display, whereas hybridomas, the single B cell platforms, the natural pairing is preserved. Another thing also to notice is about the sequence information, which is often obtained early when we are looking at the phage display or the single B cell platform, whereas with the hybridomas, we would need to sequence the hybridomas to obtain the sequence. And if we are looking at affinity, oftentimes it is really good when with hybridomas as well as the single B when compared to the phage. So at Sino Biological, we do antibody humanization as well. So this is actually a flowchart showing how the antibody humanization platform at Sino Biological works. So basically what we are doing is, let's say we have a mouse antibody. So we are identifying the CDRs and then grafting the CDRs into the human framework region, as you can see here on this figure. And then we also do back mutations, and this is like the humanized antibody, which we do express using our antibody expression platforms, and then we do validation using ELISA or other affinity platforms. So at Sino Biological, we provide antibody humanization with about 100% success rate and more than 95% sequence homology. Next, I'm going to be talking about AI-based antibody discovery service. Before getting into the AI-based antibody discovery service, I briefly wanted to talk about the history of artificial intelligence. The first digital computers came into existence in the mid-1940s, and in late 1950s is when perceptron Mark I came into existence. This is actually regarded as the first artificial neural network. In early 1990s, TD-Gammon was developed. This is a software, and it actually learned to play backgammon at a high level just below the top human players. It was in the early 2010s that AlexNet was developed. This was a pivotal early deep learning system. It was a neural network with many layers that could recognize objects such as dogs and cars at near human level. Since then, artificial intelligence, which can actually recognize language and image comparable to human, have been used in many different applications and many different areas. So at Sino Biological, we've actually partnered with A-Innocence to offer AI-powered antibody discovery service. And A-Innocence does the sequence generation part, where the sequences are generated for antibodies by A-Innocence. And at Sino Biological, what we basically do is we do the validation. So we do expression and validation of these sequences. So the program developed at A-Innocence is called Sentinels AI. So Sentinels AI is a protein design engine. It can help with lead discovery as well as lead optimization. It has multiple different platforms which could be used for different purposes. It could be used to do de novo protein design, protein-protein binding affinity modification, protein humanization, protein off-target prediction, as well as therapeutic protein developability. So the way the AI-powered antibody discovery service works is it depends on the platform which we pick. It could offer either antibody affinity maturation as well as de novo antibody design. And this is actually a schematic about how the entire process would work. So let's say if we are doing antibody affinity maturation, what we would need would be the sequence of the antigen as well as the sequence of the antibody. So once that's used as an input into the Sentinels AI database, what happens is the Sentinels AI program actually starts creating mutations in the CDR region. So the mutations could be either 1-point, 2-point, or 3-point mutations. And then the program actually starts creating a library of these mutations. And then these mutations or this mutagenesis library is then further ranked in order of its probable affinity towards the target. And the top ranked antibodies are then delivered to Sino. It could be either top 20 or top 50 or even top 100. So the top ranked antibodies, which are being transferred to Sino, at Sino Biological, what we basically do is we express these antibodies using our high throughput recombinant antibody expression platform, and then we perform validation. So often for validation, we would always suggest going about doing a primary ELISA screen, followed by secondary either BLI or SPR, depending on the need. So once we do primary ELISA, we would move the top ones to the next stage, which is the BLI or the SPR. And then the top picks from either the SPR or the BLI would then be delivered as the final deliverable for this project. With this platform, we could also do de novo antibody design. So the process would be the same, except in this case, let's say, for example, if we are looking at the de novo antibody design, we would be looking at just the antigen sequence as such. So we would just need the antigen sequence. And the Sentinels AI database actually has 100 million human antibody sequences, and the program actually screens through all the sequences, and it would find a probable antibody which could bind to this particular target. And then it would start creating mutations in the CDR, and we would go about doing the same process which I mentioned earlier for the affinity maturation. This table is just a summary showing the different project types which we could do. So for de novo antibody design, I did mention how it works. For affinity maturation, we could do either fixed template as well as template-free maturation. So when we are looking at the advantages of affinity maturation, so Sentinel's AI does offer high accuracy without any 3D structural confirmation. So over here we have the few different platforms, and you can actually see the accuracy of prediction is really high with Sentinel's AI platform. And some of the other advantages does include, shorter computation time as well as higher computational accuracy. Besides that, Sentinel's AI also offers a larger sequence space when you're taking that into consideration. Over here is a table showing the number of point mutations and the sequences. And we have 3 different platforms, and we have Sentinel's AI platform here, and you can see the comparison between the different platforms that Sentinel's AI does perform the goal at a much lower and faster rate. So for antibody development, what can artificial intelligence do? So artificial intelligence can help with designing antibodies, and it can drastically reduce the time and cost of antibody engineering processes such as antibody affinity maturation. It can also help in, de novo antibody design that I mentioned earlier. So this is just an overview slide about the different antibody development platforms that we have at Sino Biological, and some of the applications in which these antibodies could be used, and some of the functional assays which we could support. And I would like to thank everyone for listening to me, and if you guys have any questions, feel free to reach out to me. Thank you.
Hello, everybody, and welcome to our first live Q&A of the symposium. I am joined by Sumana Sundaramurthy, who is the Technical Account Manager at Sino Biological. So first of all, thank you, Sumana, for joining us.
Thank you, Sam, for the introduction.
No problem. Cool. So I'll jump straight into the questions for you, Sumana. So first of all, can the AI program used by Sino Biological and iInnocence help with off-target screening, and how would that work exactly?
Yes, of course. Thank you everyone for joining this session. And I guess if you had listened to my talk, you would have seen like the entire flowchart of how the AI program designed by iInnocence, Sentinel's AI, works. So it is somewhat similar to, I would say, both de novo design as well as antibody affinity maturation. But the goal here, the end goal here, is slightly different, where we are actually trying to screen off the targets, right? So any off-target binding, it's being screened out. So we basically, when we are trying to mutate the CDR regions of the antibody, here we'll be concentrating on reducing the off-target affinity of that particular antibody towards a specific series of off-targets, but we'll be trying to preserve the KD value or the affinity between the actual target. So a series of mutations will be created, and there'll be like a library, a mutagenesis library, similar to either the de novo design as well as the affinity maturation platform. And then the ranking would then be based on, the least affinity towards the off targets, but the preservation of the affinity towards the target will always be there. And then we will pick like the top candidates from that and then move that to Sino, where at Sino we will express those and then validate those sequences. I hope that question is answered.
Great. Thanks so much, Sumana. So moving on to our second question. What other applications can AI be used for besides antibody discovery within biotechs?
Yeah, I guess within biotech, if you're looking at antibody discovery, I did talk about, sequence design, affinity maturation. It could also be used for humanization. Other, I guess, applications could also be, process optimization in manufacturing, also for molecular stability, or even in data analysis. If we are looking in biotech, if we have a huge amount of data, if we are actually screening through a huge number of molecules, these are all applications applications in which AI could be used in biotech.
Great, thank you again. And our final question that's come through: what transformations do you anticipate in biotech owing to AI and related technologies in the next 5 to 8 years from now?
I guess it all relates to, I guess it starts with the previous question I just answered, right? Like besides antibody discovery, where else could AI be used? Data analysis. So I guess I could see it largely being used in data analysis, as well as, let's say, if we are looking in the next 5 years, it could be considered as a revolutionary age, because we are hearing about AI in like all different sectors, like healthcare, or even in diagnostics. There have been programs developed with AI where, AI is actually helping, clinicians actually diagnose cancer tumors more effectively than the actual doctors in some cases. So that could be that could bring in a great amount of change, not just in diagnostics and healthcare. We are also looking at various different other, sectors as well. But if we're looking at biotech as such, so I guess maybe the entire industry, like, almost every other player will have AI assisted project in their pipeline. So maybe, let's say, if we are not using AI at the moment, and maybe in the next 5 to 8 years, we could see at least, like I would say, anywhere between 15 to 20% of AI usage in, drug discovery, or in any stage, like at least like in the clinical stage. So I guess that's considered as a revolutionary stage, like, a stepping stone. Yeah.
Great. Thank you so much, Sumana. Really good explanation. So it's actually all the questions we've had come through. So thank you again, Sumana, for joining us online for this great Q&A session. And thanks again for your presentation.
Mm-hmm.
So that brings us to the end of this live Q&A session. We're now going to have a short break. The next presentation, will be at 2:20. So at 25 minutes. Thanks again, Sumana. Hope you have a good rest of your day.
Yeah, thank you.